Okay, so our final speaker of the morning session is Ruth Baker from Oxford University. And she will tell us about cell biology processes, model building, and validation using quantitative data.
Thanks, Stefan. And thanks also to Sam for all his organisation. I have to say this has been the easiest conference I've ever been involved in co-organising. Okay, so really just a bit of motivation. I think that if we want to, from my perspective at least, if we want to understand the kind of complex cell biology processes that are involved in development of the embryo, in repair of an organism and in disease, then we really need to go back to basics and understand them very well. And I think that although working in vitro removes clearly a lot of the a lot of important processes, we can still learn a lot from simple cell biology assays in this regard. So what I'm going to talk about today is going to be pretty much exclusively focused on a very simple cell assay that we can do in vitro, and that's a growth to confluence assay. So in this assay, what you sort of try to do, so this is an example of two different cell types. One's a breast cancer cell line and the other is a fibroblast cell line. You attempt to seed them in a dish fairly uniformly, the density is fairly uniform, and you watch as these cells move around and they proliferate and essentially grow towards a confluent monolayer. So I'm going to focus mostly on this, a very, very simple in vitro experiment, and ask what we learn about the roles of proliferation, motility, and apoptosis, so death within them, and to what extent can we quantify those processes using different types of models. But I also want to make you aware of the fact that there are different types of assays that we can do that maybe provide some different types of information. So commonly used assay is a scratch assay that you might use to look at wound healing. So you effectively grow a cell population to being more or less a confluent monolayer, and you take a pipette or something and you basically scratch away a portion of the cell, so here through the middle of this image, and then you watch as the cell population migrates in from either side of the scratch and proliferates to fill the gap. And then the third type you might think about as a barrier assay, and I think about this as in terms of almost like putting a cookie cutter down on the substrate, initializing a group of cells at a certain density within the cookie cutter, and then lifting it off and watching as the cells grow to fill the space. And you might think of this as something like a kind of the early stages of tumor growth. But I say that draw those analogies to actual kind of physiological phenomena very, very, very loosely. But I want to know what we can learn from doing these kinds of experiments. Okay, so the aim for the talk, I'm going to try to fairly quickly outline three simple models that have different levels of detail that can describe growth to confluence experiments. I'm only going to spend a very short amount of time discussing how to choose the most appropriate model, because they've got different levels of complexity, and probably we'd like to use the least complex one. That kind of becomes especially true if we want to think about our ability to infer the parameters of the model. And if I have time, I'll think about extensions to other experimental setups, but might not get that far. So this is a talk of three parts. And as such, I thought I'd try and split it up and make that very obvious. So this is a slightly random story and a bit of an aside. So I thought I'll search for some quotes that relate to the modeling part of my talk and the inference part and blah, blah, blah. So I typed into Google modeling quotes, and I just started reading them. And then I was like, oh, there's nothing about mathematical modeling here. And then it took me a few minutes to realize that was because there are other types of modeling involved in this world. But so nonetheless, I kind of stuck with the quotes that I found on my initial search, and I've put some of them into this talk. So this is the first one. I hopefully can convince you that modeling isn't a silly, crazy business, at least not in the context we're thinking about it. Okay, so my very simple model is just a discrete on-lattice model of the type that Kit talked about or outlined yesterday. So I put down a lattice on my system. It's very, very simple. I put down a lattice over the system where the lattice sites are more or less on the same sort of size as my biological cells. And I'm going to try to refer to these guys as agents, so we sort of don't get confused. And it's going to be volume excluding, so in the sense that I can have at most one agent per lattice site. And also, and I'm going to let my cells sort of basically move around, proliferate, and potentially die at different rates. So always proliferation at rate PM. I think I always take it to be 1, and scale everything else. Mix these up, haven't I? Shoot. motility at rate PM, proliferation at rate PP, and death at rate PD. Okay, and the key thing is that if a cell attempts to move, it picks one of its 4 nearest neighbor lattice sites uniformly at random, but if that lattice site is occupied by another cell, then that event is blocked. And similarly for proliferation, if this cell down here wants to proliferate, it chooses one of its nearest neighbor sites uniformly at random to put its daughter into, but if the site is already occupied by an agent, then that event is blocked. And I want to replicate these growth to confluence experiments, so I am going to initialize my domain such that each site has a certain probability of being occupied.
Sorry?
Yeah.
So the proliferation rate is proportional to the number of available sites around you, or is it constant?
So the proliferation rate is constant, but whether that event actually, so you select an agent as trying to proliferate with a constant rate, but whether the proliferation event actually takes place depends on what's going on around in the neighborhood. It's the easiest way to say it. Okay, so what can you do? Use just an event-driven algorithm to generate realizations, and from this model you can store as much information as you want. So this will come in again when it comes to talking about inference, but the idea is that I can store the position of every single agent on the lattice at every single time point. But that's a lot of information and not necessarily useful, so what we usually do is look at some summary statistic, and for example, you might think about the average agent density. So you sum up all the agents you have on the lattice at any time, and you average that over basically multiple realizations. So if you do that, you can look at how the density increases over time. So in this diagram or in this plot, there's no death, so all the lattice, every time the lattice becomes confluent. And you can plot everything in terms of a rescaled time scale down here. And what you see is that as you increase the rate of proliferation on this rescaled timescale, effectively the lattice grows to confluence more slowly. Okay, so yeah, so what really? So that's my discrete model. It's very, very basic, but I can take whatever information I want out of it. And all I'm just showing you here is effectively one summary statistic, which would be the average agent density. Don't really always like to work these kinds of models, though, because there's basically not much analysis I can do with them. So if I want to kind of look at behavior when I'm including more complex processes, maybe adhesion or death or whatever, it's very hard to do that from in any other way than just doing generating lots of realizations. So model 2 is all about basically trying to generate a mean field model of this process. So all my derivations are going to be in 1D, but they always carry over to 2D very easily. So effectively in 1D, if I have site L, then I'm looking at the rate of change of occupancy of site L, and I can just write down a conservation argument for the change in occupancy that looks something like this. So I guess it's obvious to see that sort of death events decrease occupancy, proliferation events by cells in nearest neighbor sites increase the occupancy, and then I've got some movement term that just looks like a discrete Laplacian. I can take delta, which is the lattice spacing to 0 if I want to, and by doing that I end up with just some kind of reaction diffusion equation, sort of Fisher type equation, where I've got just sort of linear diffusion and then I've got logistic growth effectively. And then if I put in my initial condition, which is all of my cells, sort of all of my lattice sites was populated initially uniformly at random, then I just wind up with effectively the logistic equation. So then I can say, well, how good does that model do? So none of this is particularly new or exciting. How does this model do at replicating the discrete data? So again, rescale everything, rescale time, rescale also the agent density, particularly important when I've got death involved, and I just get to this guy, which obviously I can solve analytically. So the key thing here is that the red in red here is the solution of this non-dimensional logistic equation, so this is my mean field model, and then the black is the comparison of all these discrete results as I vary the proliferation rate. And this is the case of no death, and you can see, just to remind you again, that as the arrow moves in this direction, that's increasing the proliferation rate. So it tells me that unless basically I have very small proliferation rates, I'm not doing a very good job of capturing the transient behavior. I always capture the steady state behavior because basically when I don't have death involved, the lattice just fills over time. But when I put death in there, things get even worse. So these are just a bunch of simulations with different proliferation and death rates. I've kept motility at 1. And what you can see is that as I up the rates, not only do I not capture the transient behavior, but I don't actually capture the steady state behavior either. And if I really kind of push this model, I can be, kind of, 10, 15% out in my steady state prediction. So what I want now is model 3, which tries to mitigate some of those issues. And what can we do with model 3? Well, what we did with model 2 kind of under the hood, and I sort of glossed over, was the fact that I was actually assuming that the occupancy of neighboring lattice sites in my model was uncorrelated. Okay, so I said things like the probability of having I see a change in the occupancy of site L because I have an agent in, for example, site L minus 1, and I have nothing in site L, and the agent in L minus 1 proliferates, and with probability of half, it drops its progeny into site L. But when I wrote down an expression like this to say that, I basically assumed that the occupancies of L minus 1 and L were uncorrelated. And if you look at just snapshots from really a sort of a simulation over time, you can see that I started this system with basically 1% of the lattice sites occupied, and they were occupied uniformly at random. There were no correlations in occupancy. But as the system kind of evolves in time, and as the agents proliferate and move around, then clearly the occupancy of sites, neighboring sites, becomes correlated. Okay. So in model 3, you tend to do something about that. So what it does is say we're going to consider the occupancy probability of single lattice sites. This is this guy here. It says that what's the probability of having the single lattice site L occupied by an agent? And then we're going to write down a conservation expression effectively for this guy. It doesn't matter about the details, but effectively just says, well, I could have an agent, I have a change in occupancy of site L, for example, because I had an agent at L minus 1. It proliferated and dropped its progeny into site L, but it was important that it was important that the occupancy probabilities of L minus 1 and L might be linked. And so this guy, row 2 here, over here, is basically just represents the occupancy of pairs of lattice sites, or the probability of pairs of lattice sites being occupied. So then I need to, if I look at this guy, I see that what I need to do then is write down expressions for how the pairwise occupancies evolve. And it all becomes a bit painful because then with these guys I have dependence on the triplet occupancy probabilities. But I can simplify things a fair bit by using kind of conservation arguments, which just relates sort of sums of maybe triplet occupancies to pairwise occupancies, and sums of pairwise to individual occupancies. But I can't do it for all the terms, and in particular, my computer's really slow, I can't do it generally for terms involving proliferation. Okay, so I need to make a closure argument, because otherwise I'm going to have a set of equations for all the individual site occupancies, another set for all the pairwise ones, and then for all the triplets and for all the and so on. And I don't want to do that. So I'm going to truncate the system using a closure approximation. The details of what it is are sort of by the by in some senses. There are lots of choices, but this one works quite well. So then I've truncated the system and I've simplified it as much as I can, and I've basically now got a system where I've got all these individual site occupancy equations, and I've got all these pair ones, and there's nothing else involved. But what else can I do? So then I need to solve them, and I need to solve them numerically. But if I can make use of two things. Firstly, I can make use of my initial condition, because all sites are occupied uniformly at random, which means that if I'm thinking about the probability of two given sites on my lattice being occupied, I don't necessarily care about where they are on the lattice, I just care what the distance is between them. Okay, so I can make a simplification using that argument. And then that also means that instead of thinking about the occupancy of single sites, I just think about the fact that every site effectively has the same probability of being occupied. That makes sense. And then I'm going to rewrite everything in terms of pairwise correlations. So essentially, pairwise correlation is just a distance L minus m is just the probability that two sites at distance L minus m are occupied divided by the individual pairwise occupancies. In short, having said a lot in a short amount of time, all I want to really highlight is that you get down to this equation for the average agent density on the lattice. And you can see that if the correlations are effectively, if there are no correlations between pairs of lattice sites, this guy would just be 1, and then back to the original mean field model I just described a minute ago. And as soon as we see correlations building up on our lattice, then effectively so F delta is the correlation between nearest neighbor lattice sites. As soon as that becomes greater than 1, then we're actually depressing the growth rates. And it goes back to what we saw. Sorry, my computer is very slow. I should point it that way too. It goes back to what we saw in the results before, that basically the population in the discrete model tends to grow less rapidly than is predicted by the mean field model, and that's because of these correlations. Okay. So what happens when you put them in and how much the results improve? So this is just showing you the same results as I showed before with both proliferation and death included, and what you can see is that we do the model does a much better job across quite a wide range of parameter space in predicting both the transient and the long-term dynamics. So really when we don't have too significant sort of proliferation and death rates, it's matching everything quite well. But even we can push the model to a place where the agents are proliferating as often as they're trying to move, and actually it still does pretty well and captures the long-term dynamics. Okay. So that's the kind of as much of the sort of modeling as I'm going to show. But to summarize, I've got a hierarchy of models that basically where model 2 and model 3, so this mean field model and this moment dynamics model, all essentially are coarse-grained descriptions of my initial very simple model 1, which is this on-lattice exclusion process model. But at some points, I guess model 2 has a simple analytic solution, but it's often quite inaccurate. And in particular, the role of motility just is gone in that model. And that model 3 is more accurate. You can do a bit of analysis, but it is pretty painful to work with. Certainly, if you want to go to 3D or if you want to include multiple populations, it's not so nice. But the last point I want to make is, and I'm not going to show any data to back this up, you have to trust me, is that both model 2 and model 3, they're usually well, model 3 in particular is relatively good at predicting the behavior of model 1, except in some parameter spaces, and model 2 is pretty hopeless at predicting the behavior of model 1 given the same parameter values. But I can find other parameter values for which the model 2 and model 3 can replicate the data of model 1. Okay, so they're not necessarily the correct parameter values, they're not what I input to model 1, but I can do it. So this begins, I guess, to motivate 2 questions. How can we tell which model is appropriate? Because if I could, I'd like to get away with model 2, because it's a logistic model, it's really easy to solve, and it's definitely very simple. But can I do that, and how do I tell when? Given I can determine what model is appropriate, to what extent can I infer the parameters of the model from data? So this is like a break between the modeling section and something talking about which model is appropriate. So this is the quote that I found. I didn't think there was life after modeling, so I've done all the modeling bit. Is the next bit going to be interesting? But maybe the next bit will pose a challenge. The only thing I had to do was cut out a bit in the middle, which didn't really go with the ethos of this talk. Okay, so can we think, really what we're asking now, given what I've just said, is are there spatial summary statistics that I can apply to my data, or in this case, my data is going to be model 1, in order to try and determine when there are significant spatial correlations appearing, and therefore direct me towards choosing whether I need model 1, this on-lattice exclusion process model, or whether I can get away with model 2 or model 3. And so one of the things we looked at was the agent coordination number. So this is effectively, if I think about the 8 nearest neighbor sites that surround any site, it's effectively the number of those sites that are occupied. So that's for any given lattice site. And if you look across the whole lattice, then that allows you to build up an agent coordination number distribution. What's the idea? Well, the idea is that if my sites were completely independent of each other in terms of their occupancy, whether there is an agent sitting in them, then essentially this will just be a binomial distribution. So what I can do is I can kind of look at data in my discrete model, or I could look at sort of my in vitro data, and I could try and work out to what extent my system looks different from this binomial distribution given a certain number of cells. And you can see kind of by eyeballing this really, so when we're in a regime where all 3 models kind of agree, the agent coordination number does look fairly much like a binomial distribution. So the green is what you would get if it was binomial distributed, and the bars are from the actual discrete model. Whereas as you begin to up the rates of proliferation and death, then it definitely departs from that. Okay. So that's one thing you can do. Relies on you putting down the lattice over actual experimental data. So maybe it's not so desirable, but it is something you could do. The other thing you could do, and it would sort of be a natural suggestion of the types of models that I've been using, is to think about the pairwise correlations and how they vary at different distances. So I think these black lines just here on the left-hand side show how in the discrete data, so in model 1, the pairwise correlations vary with time for different distances. So the arrow is basically just over 1 site, over 2 sites, and over 3 sites, I think. Okay. And then, so you can look at how the pairwise correlations vary, and then you can look at the error that's predicted by the error between your density data and your different types of models. So the error for the mean field model, the model 2 is in the red line, and the model 3 is the blue line. So then you can look at various combinations of sort of statistics that you can draw from this. So what I've shown on the right-hand side is the maximum error in the model as a function of the maximum distance between the correlations at site distance 2 and the correlation at distance 1. It's a bit of a mouthful. For a huge range of different parameter values under both the mean field model and under the moment dynamics model, so under model 2 and model 3. And you can see clearly that there's you could essentially distinguish sort of between when one model versus the other is becoming appropriate. So there are spatial summary statistics that do allow you to pick up the presence of correlations in these models. And they effectively should provide you with a quantitative measure of the accuracy of your approximate model, so of model 2 or of model 3 relative to model 1. But I think the real issue with this, and so I think this part of the talk is kind of like a negative result, is the fact that the choice of model is still going to be subjective. Because I still have to put down a threshold that says, well, when my error gets to be this much, I should switch from one model to another. And it's not clear, like, what you know, so what choice of error is going to give you what kind of accuracy. So to summarize, I guess, this part of the talk, you can determine the presence of spatial correlations, obviously using various summary statistics, but I'm not sure I figured out how they allow you to tell sort of in a non-subjective way or distinguish in a non-subjective way which models to choose. So if anyone has any suggestions in this context, I'd really like to hear them, particularly because when it comes to extending these types of models to two-species models, we really haven't found any good summary statistics that we can use to do this in a what's the word, consistent way. Okay. So now, having told you about the models and having said something about different ways to parameterize them, I want to move onto inference. And so, yeah, the world of modeling apparently can be hard. So in this context, we weren't maybe able to make as much progress as we wanted to in terms of coming up with really just good ways to distinguish which model works. This is the rest of the quote I had to chop off. But to what extent can we do some inference? So what I'm going to do in this part of the talk is really just talk about the extent to which we can sort of infer parameters of the model, and I'm only going to do that using the discrete lattice-based model. Partly because we don't have a good means of determining which model we should necessarily be using, and partly because we just haven't got that far through this work yet. Okay, so to start with, I am going to eventually show you this with some experimental data, but to start with, what I want to do is I want to generate some in silico data from model 1, and I'm going to use that to investigate the potential for parameter estimation. When I work in a Bayesian framework, I'm only going to work with cells that are motile and proliferative. For now, I'm going to forget about death or adhesion or anything else. And because the likelihood's intractable for these models, I'm going to work with approximate Bayesian computation. And the first question I'm going to have is, what summary statistics from my model are informative? So I need to do this because really what I'm doing is I'm working with big lattices, maybe 100 by 100 or 1,000 by 1,000, and I'm basically initializing them with 1% of those sites fills, and I'm watching as the lattice grows to confluence. So my data is very, very high dimensional, and I clearly can't work with that approximate Bayesian computation. So the first thing I need to do is think about what summary statistics could I use with ABC in order to try and say something about my parameter values. Okay, so just quickly, because I don't think we've had too much talk about ABC, what am I going to do? I'm going to have some prior distributions for both my motility parameter and my proliferation parameter. Effectively, there's going to be uniform distributions, and they're going to encompass kind of what we think biologically is feasible. I'm going to sample parameter values from that prior. I'm going to simulate the model using these values, parameter values, and then I'm going to evaluate how close the model output is to my in silico data using some summary statistics, so that's S, and a distance function that's lowercase d. So d star would be, in this context, I guess the data that I'm kind of trying to estimate the parameters of, and S of d would be or d would be the data that came from the model. And I'm going to repeat this process a huge number of times. I don't know, typically like, I don't know, a million or so. And then I'm going to rank my parameter sets in order of this distance function. And then basically I'm just going to take the top 1% of parameter sets, and I'm going to use them to estimate a posterior parameter distribution. Okay, and the idea is that I want to kind of do this process for a whole load of different summary statistics and try and see which of these summary statistics can tell me about which of the parameters in my model. The next slide is really busy, so apologies, but I'll hopefully take you through it fairly slowly. So I've chosen, or my student chose, 9 different summary statistics, and for each one you can generate a posterior for the parameter distribution. So just looking at this guy up here, what have we got? So here what we're saying is every time I do my ABC, sort of one round of my ABC algorithm, I basically simulate my model using the parameter values I've chosen from my prior distribution, and then I count up all the agents that are in the model with that parameter generated using those parameter values, and I evaluate how far they are away from the in silico data, or the number of cells that there was in that data. And I do that lots of times, and I rank. And what do you see? So if you put these guys all together and you look at the top 1% of parameter values, then you see that they just all fall in this band here. And the red dot, I should say, was the actual parameter values used to generate the data. So what does it tell me? Well, it tells me that if I use the number of cells as a summary statistic, then I can get a pretty good estimate of the proliferation rate within my model. But actually, I learn almost nothing about what the motility is of my cells. Okay, and it just, in this context, it seems kind of sensible. I'd understand quite a lot about the motility rates because the proliferation rate, because I am counting numbers of cells. And the reason I don't learn much about the motility is actually they started off at quite low density. And then I can repeat this then for a whole load of different summary statistics. I can look at the pairwise correlation, and you can see that I don't really gain any more information from doing that. You can look at normalized pairwise correlations, the peak height of the correlation. This is the gyration tends to the maximum and minimum eigenvalues. It just turns out that if you actually want to be able to say anything about what the motility parameter is in this context, you really have to track cells and look at either their displacement in the Manhattan sense on the grid or in the Euclidean sense. And either of those, one of those does a fairly good job. So I guess the sort of take-home message is that it looks like from this picture that we gain the most information in terms of understanding sort of distributions of potential parameter values from looking at the sort of the number of cells, the pairwise correlation, and the correlation peak heights, all basically the distance cells have moved. And that's what I just said. One of the things you can do in kind of order to quantify this is look at the KL divergence between your prior and your posterior using these different summary statistics. And it basically just highlights what I just said and what these graphs just say, that if I look at numbers of cells, things related to TD correlations, or I look at displacements, I learn a lot, and I don't learn very much from anything else. Okay. So then, to what extent can we actually apply this method to data and to the 2 different cell types that I showed you before for these for these models? Okay, so I just remind you, I've got a breast cancer cell line, these guys at the top, and I've got a fibroblast cell line. So my work with the in silico data, or our work with the in silico data, suggested that what you need to do is you need to look at the numbers of cells, and you need to look at probability tracking cells in order to in order to make progress. So we did a very, very basic sort of semi-automatic cell segmentation, put the cells down on a lattice. And then we did a very, very, very basic sort of cell tracking algorithm where we basically just tracked cells that weren't too near other cells in a very, very, very simple way. And we collected, what did we do? So we collected numbers of cells, I think, at every 6 hours, over 24 hours, and then we collected the tracks of cells, of 5 cells in any assay over, I think, 12 hours or thereabouts. And we used this as our data. And so here are the results. So it shows you sort of that we can get a fairly good estimate for the proliferation rate, both for the breast cancer cells and the fibroblast cells, by looking at the number of cells present in the assay and how that changes over time. And then if we basically are able to track cells, then we can get also fairly good estimates of the motility rate.
What are the red dots?
So the red dots are previously estimates obtained elsewhere for the same assay and for the same experiments. I should say that I think, so we were pretty consistently, I think, hitting the same parameter values. The only guy that we probably didn't do a particularly good, there are some, there's some difference for, is for the fibroblasts, and I think it's probably due to the fact that we use different cell tracking methods. But yeah, I mean, so I put them on there to highlight the fact that probably our results are consistent with some that have been obtained previously. But this guy, I'm not sure I'm too worried about the fact that we're off, because I think it's down to differences in the tracking. Okay, what do I want to say? So let's have a look at what some snapshots from these assays look like, just to more or less sort of conclude. So the fibroblasts, if we go back, we said that these 2 cell populations essentially more or less have the same proliferation rate, and actually what changes and the difference between the 2 is pretty much that the fibroblasts move a lot more quickly than the breast cancer cells do. And you can see this is sort of borne out in terms of just eyeballing the experiment. So if the red dots are sort of put down over more or less the cell centers, you can see that the fibroblasts look fairly motile, and really there aren't significant clusters forming. And that's essentially as a result of the fact that their movement rate is fairly high. But if you look at the breast cancer cell line, you can see regions of space that basically are never occupied by cells over the whole course of the simulation. So in the black squares, you can see cells that don't proliferate and just stay kind of as isolated cells over the whole time frame. And you can see guys that proliferate and cluster because they don't move very much. Okay, so this is more or less sort of as far as we've got, I would say, with this work. So it's still very much work in progress, but it looks like we can at least isolate summary statistics that allow us to make predictions of the parameter values for these types of models. Okay, and that was all for model 1. So I just want to, I guess I've got a few minutes still to go. I just want to finish up by maybe trying to say something about what this means for inference in the other models. So for these growth to confluence experiments, where I put down everything, try to put my cells down uniformly at random, I need to get individual cell tracks as well as sort of to count cell numbers in order to estimate both the motility parameter and the proliferation parameter. These data are naturally available in Model 1, right? They're very easy summary statistics to get out, so it makes Model 1 sort of a very good choice to use, I think, if you definitely need cell tracks to infer, in this case, the motility parameter. But it is possible, and we've done a bit of work on this, to construct a model for mean cell trajectories from either Model 2 or Model 3. And so then maybe in that context we could think about parameterizing Model 2 and Model 3 using these summary statistics if we needed to. I think that was the point I wanted to make. But the other thing you could do is to change your experiment, right? So the reason that we really needed to track cells in these experiments is because they started off at initially quite low densities, and basically there were really no significant enough pairwise correlations to allow us to get a handle on both the movement and the proliferation rate from basically from the correlations. So can you change your experiment, and does that allow you to do something different and do it more easily? Well, actually, what we then went to do was to look at scratch assays and see, well, if our cells are kind of naturally got correlations in space, spatial correlations, does that allow us to estimate both the parameters in our model without necessarily having to track cells? And I guess the answer is that I think to some extent you can do that in this case. So looking at the number of cells again really only gives you an estimate of the proliferation rate, but if you put that together with the pairwise correlation in this context, I think you can do a fairly good job at getting both the proliferation rate and the motility rate out of the same experiment. And the real key thing about this is it doesn't rely on your ability to track cells, and tracking cells is really painful, right? So often, especially for the fibroblast population, we really had to do it by hand because they've got quite funny, they're not just kind of little round blobs that just sit there, right? They're quite hard to segment, they're quite hard to track. So if you can design an experiment that allows you to get data out without tracking cells, that really is the kind of thing that you want to be doing. Okay, what else do I have to say? Yeah, so in summary, so we've outlined a range of models at different levels of resolution, effectively making more or fewer assumptions to describe really the dynamics of motile proliferative cell populations, but it's very easy to add in death and it's easy to add in adhesion as well. I've tried to outline the extent to which I think it's possible to determine which of these models is appropriate given some data, and it's demonstrated that it is possible to infer model parameters for a growth to confluence experiment given I have certain summary statistics. So growth to confluence experiments are very simple in the sense that all the kind of the coarse-grained models, say model 2 and model 3 that you write down, are very, very simple. But it is possible to repeat all this kind of this process for experiments with non-uniform initial conditions. And I think one of the reasons you might want to do that is that it mitigates the need for cell tracking, which is a fairly time-consuming and difficult process. Okay, so yeah, I think that's everything I wanted to say. So I just want to thank three people in particular. So Matt Simpson is a long-standing collaborator at QUT, and we started off doing this kind of mean field approximation stuff in 2010. Mm-hmm. And then Debbie Markham and Andrew Parker were basically the people who've done all the work on the modelling and the ABC stuff too. So there are students in Oxford who've done a great job on this. There's a few references, there are others, but I'm happy to chat about them, and I'm happy to answer any questions if you have any.
Yes, question?
Over there. If I understood correctly, you were saying that when you go between your fine-grained model 1 and the coarse-grained models, you can get them to agree, but the parameters in your coarse-grained model are not sort of obviously related to the one in the fine-grained.
Yeah,
do you have an idea of how to, what the mapping of good coarse-grained parameters to fine-grained parameters is?
Sorry, I'm not sure I quite understand. So you're asking...
is there a principled way to say, if I want to, just forget about experiment, I'm just asking about a model.
Yeah.
If I want to sort of make model 2 or model 3 replicate model 1, have you either empirically or in a principled way figured out how to extract from the model 1 parameters
Yeah. No, I guess is the short answer. We've looked at the extent to which the estimates would vary. So, for example, for the data that I showed you, so with actually the data, I think if you use the logistic model as opposed to the discrete model, for the proliferation rate, you can be off by something like 20 or 30%. So it clearly is you're clearly kind of missing it by quite a long way. Have we looked at systematically what the kind of mapping is? No, and I don't know. I'm not. Maybe have suggestions. I'm not sure how much or what we'd learn necessarily by doing it, and that's maybe why. But
similar issue. That is some curious.
Yeah. Okay. Cool.
More questions?
I'll ask a question then. So this T=0 hours, the breast cancer, it almost looks like it's clustered on deposition. And,
Yeah,
to what extent does that screw up your parameter estimation?
Yeah, maybe a bit. This one definitely looks like it is. I'm not.
Like I know these cells are often clumped when they.
It's quite hard to pin them down randomly.
Mix them a lot.
Yeah, I mean, I think I can't tell you the numbers off the top of my head. I don't think it comes up as being terribly significant in terms of relative to this kind of case, but clearly, yes, it would play a role. And I think if you were going to do this kind of parameter estimation properly, you would have to use Model 3, and you would have to feed it directly the kind of actual initial conditions you see here, so that if you wanted to do a good job. If you went with a discrete model, it doesn't matter too much, because literally what we do is basically impose a grid on here and assign one cell to every lattice site. So we kind of inbuilt, you start with the right, quote unquote, the right initial conditions anyway, if you like. So that's not a problem. Clearly you can't do that for the logistic model because they're not there. But yeah, it is a good point, and I think it's something we should be careful to take into account.
More questions? Otherwise, let's thank the speaker and have lunch.